Synthesis of progesterone. Pregnenolone serves as a principal precursor to all other steroid hormones synthesized by the ovary, testes, or adrenals. It appears that the rate-limiting step for the synthesis of progesterone is side chain cleavage of cholesterol by 450 SEC. Pregnenolone is then converted into progesterone by 3-beta hydroxysteroid dehydrogenase, that is abbreviated as 3-BHST. Thus, deficiencies in either P450-SEC or 3-BHSD have profound effects on the synthesis of all steroids. In the ovary, progesterone is produced at all stages of follicular development as an intermediate for androgens and estrogen synthesis, but becomes a primary secretory product during the peri and post ovulatory luteal phases. The synthesis of progesterone is under the control of FSH during the early stages of folliculogenesis, and following acquisition of LH receptors becomes sensitive to LH later in the ovarian cycle. The synthesis of progesterone by the corpus luteum is stimulated during early pregnancy by increasing levels of chorionic gonadotropin. In addition, the placenta secretes high levels of progesterone during pregnancy, although a different isozyme of 3-BHSD is involved for this synthesis. You can see over here the basic in vitro study for the preparation of progesterone. The starting material which is used for the synthesis of progesterone is diosgenin. The diosgenin molecule consists of basic structures of the steroid and tetrahydrofuran and sugar moiety at carbon 17 position, and hydroxyl group at carbon 3 positions, as well as double bond at carbon 5 position. So this molecule is treated with pyridine in the presence of hydrochloric acid, and the proton which is present at carbon 3 position with the oxygen atom is converted into acetyl moiety. So the second intermediate is further treated with chromium trioxide, which is used for the cleavage of tetrahydrofuran moiety, which is present at carbon 17, means 4 member rings of the basic structure of the steroid molecules, and in this way the ruptures of the 6 member ring of tetrahydrofuran moiety is carried out. This intermediate is further treated with acetyl alcohol, and which is used for the removal of sugar moiety. In this way, we obtain an acetyl group at carbon 17 positions of the basic structures of steroid molecules. Now, the double bond which is present at carbon 17 position is reduced by using hydrogen in the presence of platinum, which is the general method for the reductions of the double bond. And after that, we will get the next molecule. The next molecule is that in which acetyl group is present at carbon 3 positions, double bond is present at carbon 5 position, and acetyl group is present at carbon 17 position. This is the second last molecule, which is further treated with in the first step with alkali, that is potassium hydroxide, and in the second step with chromium trioxide. So the acetyl group is further reduced into carbonyl group at carbon 3 position. In this way, we will get the final product, that is progesterone, in which carbonyl group is present at carbon 3 positions and double bond is present at carbon 4 positions, as well as acetyl group is present at carbon 317 positions by the additions of 2 methyl groups. So this is the general mechanism for the synthesis of progesterone, which is known as in vitro study of the whole procedure. Let's see the synthesis of androgens. Androgens are synthesized and secreted primarily by the Leydig cells of the testes, thecal cells of the ovary, and cells in the reticularis regions of the adrenals. In most tetrapod vertebrates, testosterone is the dominant circulating androgens. Testicular synthesis and secretions of the testosterone is stimulated by circulating LH, which upregulates the amount of 17-alpha hydroxylase, that is carbon 17 carbon 20 lyase enzyme, a rate limiting enzyme for convergence of carbon 21 into carbon 19 steroids. Once taken up by target tissues, testosterone can be reduced by 5-alpha reductase to shield a more active androgens metabolite, 5-alpha dihydrotestosterone, that is generally represented as 5-A-DHT. Testosterone and androstenedione can also be converted into estrogens, such as 17 beta estradiol, that is E2, or estrone through a process termed aromatization. Aromatization is carried out by a cytochrome, that is P450 aromatase enzyme, that is expressed in the granulosa cells of the ovary, lighting cells of the testes, and many other tissues, including the placenta, brain, pituitary glands, liver, and adipose tissues. Indeed, many of the effects of circulating testosterone are owing to conversions into either 5-alpha dihydrotestosterone or E2 within the target tissues. Let's see the synthesis of testosterone, because testosterone falls in androgens, because this is the big examples of androgen steroids. So let's see the first step, in which 17 hydroxypregnenolone is converted into dehydroepiandrosterone in the presence of 17 alpha hydroxylase 1720 lyase enzymes. So you can see over here in the first reaction, you can see the structure of 17 hydroxy pregnenolone, in which the hydroxyl group is present at carbon 3 position and acetyl group is present at carbon 17 position. In addition, one hydroxyl group is present as alpha in the 17 position too. So this structure is converted into dehydroepiandrosterone in the presence of enzyme 17 alpha hydroxylase 1720 and lyase. The structures of dehydroepiandrosterone consist of two functional moieties. One is hydroxyl group at carbon 3 position and the other is carbonyl group at carbon 17 positions by the additions of double bond at carbon 4 position. So when we see the difference between the structures of these two molecules, you can see over here there is a presence of just 17 positions carbonyl and hydroxyl group at carbon 3 positions and double bond at carbon 4 position in the first intermediate molecules. In the second step, dehydroepiandrosterone is converted into androstenedione in the presence of 3-beta hydroxysteroids dehydrogenase enzyme. So the androstenedione consists of the carbonyl group at carbon 3 position and double bond at carbon 4 positions. In addition, carbonyl group at carbon 17 position. So just only the changes that takes place at carbon 3 positions is the presence of carbonyl group at carbon 3 positions in the androstenedione instead of hydroxyl group, which was present in the previous structures in dehydroepiandrosterone. In third step, androstenedione is converted into testosterone, which is our target compound for the synthesis of androgens or testosterone in the presence of 17-beta hydroxysteroid dehydrogenase enzyme, which is abbreviated as 17-BHSD. So you can see in this reaction, the androstenedione is converted in testosterone. In testosterone, there is a presence of carbonyl group at carbon 3 positions and double bond at carbon 4 positions, similar as that of androstenedione. But the carbonyl group, which was present at carbon 17 position, is converted into hydroxyl group as beta. So the difference between testosterone and androstenedione is just there is the presence of hydroxyl group at carbon 17 position in testosterone as compared to carbonyl group at 17 positions in androstenedione. So this is the general method for the synthesis of testosterone. Synthesis of estrogens. Estrogens and progestins are synthesized and secreted primarily by maturing follicles, corpora lutea of ovaries, and the placenta during pregnancy. The predominant secreted is E2 and the predominant progestins is progesterones. The profile of the synthesis of estrogen changes during the course of folliculogenesis, during which, under the influence of LH, the thecal cells synthesized and secrete androstenedione and testosterone, which diffuse across the basement membrane and are subsequently aromatized to estrone and E2, respectively, by the granulosa cells. The level of aromatase and hence estrogens produced in the granulosa cells is under the control of FSH during mid-follicular phases. Later in the cycle, the follicle corpora lutea express greater numbers of LH receptors and LH begins to regulate E2 production. During pregnancy, the placenta utilizes androgens precursors from the fetal adrenal glands and secretes large amount of E2. In addition, in male vertebrates, many target tissues such as pituitary cells and hypothalamic neurons convert circulating testosterone into E2 steroids. Let's see the synthesis of estradiol. Estradiol is the common example of estrogens. In the first step, 11-hydroxypregnenolone is converted into dehydroepiandrosterone in the presence of 17-alpha hydroxylase 1720 ligase enzyme, which is then converted into androstenedione in the presence of 3-beta hydroxysteroid dehydrogenase. So you can see over here in this reaction, the first structure is the 7-hydroxypregnenolone, which have hydroxyl group at carbon 3 position, acetyl group at carbon 17 positions, and hydroxyl group as alpha also present at carbon 17 position. In addition, there is a presence of double bond at carbon 5 position. This molecule is converted into dehydroepiandrosterone, in which the acetyl group and the hydroxyl group, which is present at carbon 17 position in the previous molecule, is converted into carbonyl group. So this molecule is known as dehydroepiandrosterone, which is further converted into the next molecule by the effect of enzyme 3-BHSD. In second step, androstenedione is converted into estrone in the presence of aromatase enzyme, which is abbreviated as CYP19. Androstenedione is converted into estrone. You can see in this reaction, the difference between these two structures is the presence of first aromatic ring in the estrone, which is not present in the previous molecule. So there is also a change at carbon 3 positions because the carbonyl group is present in the androstenedione. However, there is a presence of hydroxyl group at carbon 3 position in the estrone. In the third step, the estrone is rearranged into estradiol in the presence of 3-beta hydroxysteroid dehydrogenase, which is abbreviated as 17-BHSD. The estrone, which has aromatic ring in the steroidal structure, which is a first ring which have the hydroxyl group at carbon 3 positions and carbonyl group at carbon 17 position, is converted into estradiol. In estradiol, there is a presence of 2 hydroxyl group. As the name implies, diol, one hydroxyl group is present at carbon 3 positions and the other is present at carbon 17 position. In this way, the estradiol is synthesized by using this reaction scheme.